All right. All right, so, oh, that's not where I want to start. There we go. So, as I promised, we're going to spend a lot of time over the next couple weeks or so talking about neuro. And we'll start, again, as I promised, within this particular unit and within this particular system, talking about sort of the fundamental sort of structure and function of the system. We'll move on then talking more about the actual conditions and diseases and other sort of bodily control that the neurologic system has on our bodies, as well as we start to talk throughout the next few days. But today will be structure and function. And again, hopefully will not just be straight structure and function, but we'll be adding some clinical sort of, nuggets, so to speak, into this talk as well to make it sort of come alive for you today. So here are your objectives. Again, exact same as last semester. Do what you did with the objectives. However you use the objectives to prep and study and use as your study guide for the exam, do it again. Nothing's changing there. Expectations will be the exact same in terms of the objectives and how you sort of focus your reading and your preparation. And there's a lot of them for neuro. So let's get started. So overview of the neurologic system. So we know, right, from your anatomy and physiology courses and maybe even a little bit of your biology courses, that the nervous system is basically divided into two major systems, right? The central nervous system and the peripheral nervous system. Now we know that the central nervous system contains of basically just two things, the brain and the spinal cord. And we know that the peripheral nervous system contains all the other things in the periphery. So things that you'll see, so cranial nerves, spinal nerves, and then there are two specific sort of pathways within the peripheral nervous system that I do want to spend a little bit of information or a little bit of time talking about that information here on this screen. So let's make, again, for some of you today, this will be a really nice review. For others, it may feel like, oh, good, I need to brush up a little bit. It's been a while since I've thought about some of these things and some of these systems. So either way, hopefully today will be purposeful for you all. So let's talk a little bit about the pathways. So we have an afferent, so afferent, ascending pathway in our body, in our nervous system, and we have an efferent, or a descending pathway in the spinal cord, between the sort of brain and spinal cord in our nervous system. Let me ask you this. Here's our review. We start now. What kind of information is carried on the afferent, ascending pathway? What do you think? Sensory. Right. So it's carrying, so it moves things from bottom up to the brain, so ascending, afferent. It takes those things that we learn about in our environment, through our senses, through our touch, and takes that information from the environment, from our fingertips, from our muscle, from our skin, wherever we're sensing something, and moves it up, 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 up to the brain. So that's afferent. So carry impulses to the brain, sensory, and sensory information usually comes into the spinal cord, so from the peripheral nerves into the spinal cord in what location? Do we know? More toward the front, so the ventral side, or the back, the dorsal side of the spinal cord? Anyone remember? Dorsal. Yeah, dorsal. Dorsal root, dorsal column. When you hear those terms, in terms of the actual geography and sort of anatomy of the spinal cord, the sensory information comes into the spinal cord from our periphery, from our peripheral nerves, into the dorsal aspect of the spinal cord. So here you see it right here. This is my lovely cross-section of the spinal cord, right? Here's the ventral side, the front side. Here's the back side, the dorsal side. And it's this sensory section right back here that I've sort of highlighted here. That's our dorsal column that feels touch, pressure, proprioception, vibration. That's the big stuff. That's the big sensory stuff that the dorsal side, the dorsal column of the spinal cord senses. We're going to talk about the difference between the gray matter and the white matter in the spinal cord in a second. Yeah, let's hold on that. I don't want to get confused. We'll talk about that in a little bit. Okay. Good. Okay, so how about the efferent? How about the descending path from the brain to the spinal cord out to the periphery? What kind of information is carried on the efferent or descending pathway? The opposite, motor. Right, exactly. So it innervates the skeletal muscles. This information comes from the brain to the spinal cord out to the peripheral nerves. It carries information to innervate skeletal muscles, and it also carries information to innervate effector muscles or effector organs. What do I mean by effector? when you hear that term, effector? Well, involuntarily, our organs are also sort of controlled by our nervous system, right? There's a lot of nervous system control of our organs in terms of keeping our body in our dynamic, steady state. So our organs are controlled by the nervous system as well, and that's also fed through that efferent descending pathway. Good. So when you think of afferent, think of sensory, specifically through that dorsal root or dorsal column. When you think of efferent pathways, think both voluntary and involuntary control of our muscles and our organs. Good. Any questions about that? Okay. Okay, so now we're going to go a little bit deeper, even more, into that peripheral nervous system. Functionally, the peripheral nervous system can be divided into the somatic nervous system and the autonomic nervous system. So the somatic nervous system carries our motor and our sensory pathways that we just sort of talked about, regulating more voluntary motor control of the skeletal muscles. So again, when you think of somatic nervous system, and the word somatic means what? Bodily, right. So when you think of somatic nervous system, think voluntary. This is voluntary control of skeletal muscles, period. When you hear autonomic nervous system, remember the autonomic nervous system, again, has motor and sensory pathways regulating the body's internal environment through involuntary control of organ systems, right? So when we think autonomic nervous system, think involuntary. So this is not our brain telling our bicep to contract anymore. This is telling our pancreas to secrete amylase, right? So it's not a voluntary process. That is autonomic control throughout the nervous system. Does that make sense? Do you see the difference there? And then, to take it even one more step further, within the autonomic nervous system, you have the sympathetic branch and you have the parasympathetic branch. And we will talk later, if we don't get to it today, we'll talk about more specifically the functions of the sympathetic and the parasympathetic nervous system, either later today or on Monday. So I just wanted to sort of keep that in mind, okay? So the nervous system can be branched very specifically. You start big with central and peripheral. Then with peripheral, you can branch down even farther into somatic and autonomic. Then within somatic and autonomic, you can branch even further in autonomic to sympathetic and parasympathetic. And actually, if you want to flip, farther on through the lecture, I actually have a really good slide, sort of a summary of what I just said in terms of the branches. So that's there for you visually as well. Okay, so that was a quick overview of the branches. We're going to get back to more specifics of that in just a minute. Soon we'll talk about all the roles and sort of functions of these different branches. But first, I want to look at sort of a micro level of the nervous system to sort of start with and start there together, and then move forward to sort of the macro level, talking about the branches more specifically and their functions. Okay, so again, we're sort of moving away from the branches for a minute, and we're going to talk on more of a micro level. So you can't talk about the neurologic system without talking about the neuron, right? The neuron is the primary cell of the nervous system. Is it the only cell? Is it the only type of cell found in the neurologic system? No. But it's the primary cell of the nervous system. Whereas there's other cells in the nervous system, like neuroglial cells, which you'll see you'll find neuroglial cells in the central nervous system, or Schwann cells in the peripheral nervous system. But either way, no matter if you're talking about either the neuroglial cells or the Schwann cells, they both provide sort of structural support and nutrition and those types of sort of supporting cast of character type roles for the neuron. The neuron is the primary cell in which, the neurologic system is sort of based upon. We know, right, depending on where the neuron is and what it's innervating and what it's working on, that it varies in size and structure. It has several different cellular constituents, which we are not going to spend a lot of time on. They're just up there on the board for your information. The two that we may sort of end up talking about or touching on a little bit in terms of the cellular constituents are the neurofibrils, which are the responsible for transport of the cellular products within the neurologic system, and something called Nissl substances, which are involved in the protein synthesis in the neurologic system. But again, we're not going to get really hung up on some of these cellular constituents. And you know this already, but some neurons, like olfactory neurons, continue to divide, but we know that that's not a normality, right, for neurologic cells, for neurons. Especially in the central nervous system, if neurons die or neurons are injured, that's usually a permanent effect. Specifically talking about this in the central nervous system, like brain and spinal cord. We can sort of talk about some regeneration and some healing if we're talking about neurons in the peripheral nervous system, which we'll get to. Okay. So either working alone or working in units, neurons affect environmental changes in our bodies. They initiate body responses to something in our environment that our body senses through our sensory system. And they do all of this to sort of maintain that healthy, normal, dynamic, steady state in our body. So the structure, as I mentioned already, can vary markedly depending on neuron to neuron. Each neuron is adapted to sort of perform specialized functions. But no matter what the structure, no matter what the function, all neurons have three main components, right? And you see those here on the screen. They are made up of the cell body or the soma. They have a dendrite or dendrites, and they have axons. So the cell body is located mainly in the central nervous system. So basically, when we're talking about neurons, the cell body of a neuron you'll find in the central nervous system, which means where would you find a neuron? The actual cell body of a neuron? In the brain or the, good. Dendrites, which are the what? What do dendrites do? What's their major function? Again, this is a review. What's their major function in terms of their role for the neuron? They're the receivers, right. They are the receivers. They're the receptive portion of the neuron. Their extensions then carry those nerve impulses that they have received toward the cell body, toward the soma. And then once the nerve impulse or that information reaches the cell body or reaches the soma, what is in charge of conducting that information away from the soma to the next neuron? Axon. Good. So remember the axon carries things way, right? A's. Dendrites carry to, axon carries away. They're long conductive projections from the cell body that carry nerve impulses away from the cell body. Good. And here's a great diagram of the neuron. Specifically, we're going to be looking at this diagram as we talk about the next slide, which talks more specifically about some of the important roles and functions of the axon. So I'm actually going to stay on this screen and then move, in terms of what I'm going to be talking about, we'll move forward to the next slide, which talks more specifically about the parts of the axon. So axons carry nerve impulses away from the cell body. Yes, we said that. Here we see the dendrites, which are picking up information. And what are dendrites usually, what is it that they're picking up from other neurons? What is the actual information that they're receiving? In terms of, what are they actually picking up on the dendritic tips? Neurotransmitters, right? That got sent across the synapse from a previous neuron. So the dendrites are picking up the neurotransmitters that have been sent across that synapse from another neuron. It brings that information down to the soma, to the cell body, and then from there, the information is sent on down the axon. So the first area of the axon is this sort of conical shape right here of the axon called the axon hillock. The axon hillock is really interesting. Not only is it sort of, interesting in terms of its shape, but this is the initial segment of the axon that is very, very sort of desensitized. I shouldn't say desensitized. It's actually very sensitive to stimulation, to an action potential. So here is where an action potential actually starts, right? We talked about action potentials last semester, and that sort of cardiac nerves and neuronerves are the ones that can really sort of be excitable in terms of action potentials, and this is where it begins. This is the part of the nerve cell that's really excitable and really ready to receive an action potential, the axon hillock. From there, the information gets sent down the axon. So now you can go to the next slide that looks like this, and we'll talk about this slide as we're looking at the diagram. Does that make sense? You guys following me? I just think it's easier to look at the picture while we're talking. Okay, so what do you see here covering the axon? Schwann cells. And what are the Schwann cells covering? Good, the myelin sheath. Right, exactly. So the myelin, a typical neuron has only one axon, which may be covered with that segmented lipid layer of lipid material called the myelin sheath. And the cell membrane, most specifically, of a myelin cell or a myelin sheath is the Schwann cell. So good, you were both right. So each myelin sheath is covered externally by the cell membrane, which is a Schwann cell. Good. So myelin is a segmented layer of lipid material surrounding axons. What is the beauty of the myelin sheath? Why are we so hung up on the function of what this myelin does? What does it do for the cell? Right, so it's an insulating substance that speeds up the transmission down the axon. So the sort of lay example I always give to sort of like patients when I'm trying to explain this is the myelin sheath is basically the rubber coating on the outside of an electrical cord, right? What happens if something wears away or if that rubber coating around an electrical cord, let's say on a lamp, is damaged? What happens in terms of that electrical impulse going down that wire to the lamp? What happens to the lamp's function? Right, so the electricity can sort of escape. It doesn't conduct it down the axon, yeah? Yes, it's a demyelinating disorder, which is MS, and we're going to talk about that later. But the myelin sheath is basically a conducting agent that sort of insulates the axon, or in the example of an electrical cord on a lamp, the actual electrical wire. And it allows the electricity to be shot down efficiently down that wire, aka axon, and produce some sort of function on the other end. So in the example of the lamp, for the light to turn on. In the example of a nerve innervating my bicep, the bicep to contract. So very good. So when you think of a myelin sheath, think of an insulating sort of electrical cord example. Does that make sense? Okay. It's formed and maintained by those Schwann cells. They are the cell membranes of a myelin sheath. Good. Yeah, that's what I wanted to say about that. I wanted to make sure I got everything that I wanted to sort of mention about that. Any questions about the axon versus the Schwann cell and sort of and the myelin sheath and their sort of relationship together? Okay. Then moving on, axons carry the nerve impulses away from the cell body. Oops, there we go. We're on this slide now. So now, I'm going to go back to the picture just so you sort of know where I am. Now you have something in between. So the myelin sheath, as you can see here from this picture, does not run continuously down the axon. You see how there's different myelin sheath segments, right? Each one of these little areas in between the myelin sheaths are called nodes of Ranvier. Nodes of Ranvier. They're the regular sort of interruptions of the myelin sheath. And it's here at these nodes of Ranvier are really the only places along the axon that an axon can branch. So you can see here where they actually, there's sort of axon collateral sort of branching off here from a node of Ranvier. Now, in terms of the sort of role or the function of these myelin sheaths, we know they help conduct. But there's a specific type of conduction that occurs along the axon because of myelin sheaths, and that's something called saltatory conduction. Does anyone remember that, or anyone know what that means exactly in terms of how it conducts down that axon? Okay, so it actually makes conduction more efficient. So the reason, the way it does is that the flow of ions between segments of the myelin sheath sort of flow from sheath to sheath, so myelin sheath to myelin sheath, instead of trying to go all the way down the myelin sheath at one time. So do you think that makes it more efficient or less efficient in the way that the ions sort of hop from myelin sheath to myelin sheath? Is that good or bad? Do we want that? Yes. It actually speeds up the velocity. It actually makes that electrical impulse down an axon go faster because the ions can just jump all the way down the axon from myelin sheath to myelin sheath instead of trying to go all the way down the axon at one time. It actually makes conduction a lot more efficient, and actually it makes it a lot more fast. It's a lot faster. Speeds up the velocity. Good. All right. There's also two more sort of vocabulary words that I would like to discuss when talking about the axon, and that is divergence versus convergence. So divergence is the ability of an axon, of a branching axon, to influence many different neurons. And I actually have a really, simplistic but clear diagram up here of one neuron who, because of its branches, maybe at those branches of nodes of Ranvier, branch out. The axon actually branches out and innervates several different neurons instead of just one neuron impacting one neuron. That is called divergence. Okay? So this, talking to three neurons, is called divergence. Now, you can have the opposite. You can have branches of numerous neurons converging on one or a few neurons. So a whole bunch of neurons coming down and talking to and innervating one other neuron, so communicating with only one neuron. And that is called convergence, and that's what's happening here. Does that make sense? Okay, so we just spent a lot of time talking about specifically axons on, as part of a neuron, of a part of a nerve cell. Why? Why do we care so much about the axons? And why are axons such a big deal? And we actually talked about it just a second ago when we talked about demyelinating diseases. And a perfect example of a demyelinating disease is multiple sclerosis. So disorders of the myelin sheath, which are called demyelinating diseases, such as MS, or another disorder you might have heard of called Guillain-Barré syndrome, really does show and highlight the importance of that myelin sheath. Because if there is any type of either disruption or complete sort of destruction of a myelin sheath, that impulse trying to reach and go down that axon and reach a muscle fiber or reach an organ or whatever it may be is either going to it's going to slow down the conduction. So let's say, for a simplistic example, we're talking about, moving my arm, moving my bicep, right? If there is some type of disruption to that myelin sheath of the axon that's trying to innervate my bicep, I'm not going to really be able to contract my bicep in a sort of efficient way. So it might be slowed, my bicep contraction might be weaker. And then if it gets to be so bad that the myelin sheath has been completely destroyed and there really is no myelin sheath on that axon to speak of anymore, then I'm going to have complete stoppage of function. I'm going to have paralysis. I'm going to have complete lack of use of that bicep. Does that make sense? That's why we're so concerned about axons and the health of the myelin sheath and the Schwann cells around that axon. Good. All right. Any questions about that? As we're going to leave neuron and sort of talk more about the supporting cast of characters, the other nerve cells in the neurologic system. Is that clear? Okay. All right. We also, when we're talking about neurons, need to talk a little bit about the functional classification of neurons. I talked about how there are different functions and different sort of ways in which neurons get used, so to speak, in our neurologic system. And those basically just get classified into sensory, motor, or associational. So sensory neurons are obviously the neurons that transmit impulses from the sensory receptors in our peripheral nervous system, so on our fingertips, in our skin, in our muscles, and carries that information up to the central nervous system from the brain or the spinal cord up to the brain. Motor obviously is the opposite, so it transmits impulses from the central nervous system, so from the brain and spinal cord out to an effector organ or an effector muscle. But then we also have associational neurons who really just transmit information from neuron to neuron. So they're not they're kind of the in-between, the go-between, so to speak. They're not necessarily sensory, they're not necessarily motor. They're somewhere in the middle, having those two systems sort of talk to each other. Does that make sense? Okay. So functionally, there are 3 types of neurons, is what I want you to carry away from that slide. Okay, now I did mention there's some other sort of supporting cast of characters that we can't sort of leave behind when we're talking about the neurologic system. One of them is called neuroglia. We talk very sort of in layman's terms about neuroglia being sort of the nerve glue of the nervous system. They support the neurons of the central nervous system, and there can be several different types of neuroglia with several different types of functions. So we have astrocytes that sort of fill the space between the neurons and surround the blood vessels within the central nervous system. Then we have another type of neuroglia called oligodendrocytes. Now these are the cells that actually deposit the myelin sheaths or help sort of create, have a relationship with those Schwann cells to create the myelin sheaths within the nervous system. Again, in these demyelinating disorders that we talked about, like multiple sclerosis, there's a major disturbance in the oligodendrocytes because they're not laying down the appropriate myelin sheath. They're not creating the appropriate myelin sheath around that axon. So you'll quite commonly see a disruption in the oligodendrocytes in a disease like MS. And then microglia, basically just sort of the cleanup guys. They sort of remove debris through phagocytosis within the central nervous system. Okay, any questions about that? Sort of supporting cast. Not necessarily a neuron, but still a nerve cell in the neurologic system. Any questions about those? Okay. So it sort of makes sense now, as we're talking about structure and function on sort of a micro level of the neurologic system, that we talk about what happens when an axon is damaged or when an axon is completely severed. What happens when a nerve, or I shouldn't say a nerve cell, but specifically the axon of a nerve cell is damaged is something called Wallerian degeneration. So Wallerian degeneration, as you see here on the screen, is degeneration of a nerve fiber that's been separated from its nutritive source, from the cell body, from the soma, either by injury or disease. And it occurs, and I want to be clear here, Wallerian degeneration occurs distally on the axon, distal to the injury. So let's say if this, here's my lovely neuron, right? My cell body. And here's the axon branching off. If, let's say, there's an injury or a cut right here on this axon, Wallerian degeneration, I'm going to abbreviate WD, would occur here distal, the part that's been cut off from the rest of the cell. Does that make sense? And so what you'll see distal to the injury on that axon is that you'll see actual swelling, so cellular swelling appears to that part of the axon. Some of those neurofilaments, those things that we talked about earlier, sort of actually hypertrophy. And it makes sense that they hypertrophy. They're not getting communication and nutrition. The myelin sheath and this part of the axon would start to also hypertrophy, or atrophy. And sort of degenerate, the myelin sheath along that part of the axon. The axon portion also sort of degenerates and disappears. Now, interestingly though, the myelin sheath can reform sort of through its Schwann cells, through its cell membranes, right? And it can align, the Schwann cells can sort of align in a column between the severed part of the axon and whatever organ that it's affecting. So let's say this is the tips of the finger, right? Here's the muscles and the tips of the finger. In this area between the actual axon injury and the effector muscle that it's trying to affect, Schwann cells can actually sort of, even though this, let's say this axon's kind of completely gone, it's kind of been degenerated, Schwann cells can sort of realign along where the shaft of that axon should be. And if the axon does regenerate, it sort of, the Schwann cells sort of act as sort of this guiding tunnel in which the axon kind of regrows through that tunnel of Schwann cells, through that myelin sheath tunnel, so to speak. So that's Wallerian degeneration. Now, what happens proximal to the injury? So we talked about Wallerian degeneration, what goes on from injury distally. What happens from injury and then proximal to that cell body, to that nerve cell in general? So swelling and dispersal of those nissl substances, we talked about that a little bit earlier. Cell increases in metabolic activity, in protein synthesis, and mitochondrial activity. So very similar to what would happen in any other type of sort of injury or inflammatory process in our body. And if it's going to sprout, if it's going to regenerate, new terminal sprouts project from this proximal segment within 7 to 14 days. So again, let's say the axon's been cut right here. New little sprouts, if it's going to sprout, will start to actually regrow here from this, from this little proximal area where it was cut within 7 to 14 days. Now, to be clear, mature nerve cells that we're going to find in the brain or the spinal cord do not divide, right? And injury can cause permanent loss of function. And so the perfect example of that would be like a spinal cord injury. This does not happen. Wallerian degeneration and sort of the potential for axonal regrowth does not happen in the central nervous system. You're not going to see this in a spinal cord injury. This is more the type of sort of regeneration that you might see in an injury to the peripheral nerve. So in a finger, in a toe, in somewhere out in the periphery. The greater chance of regeneration is in the peripheral nervous system. I want to be clear about that. Also, interestingly, the closer to the cell body of the nerve that this injury has occurred, the worse the prognosis in terms of its ability to sort of be able to regenerate. So the closer down here to the effector muscle that this axon's been severed, the better chance of axonal regrowth than if it got severed way up here by the soma, just sort of interestingly. So yes, the process is limited to myelinated axons, so generally only in the peripheral nervous system. Depends on location, type of injury, the presence of inflammatory response, and scar tissue formation in that area. Again, it should make sort of intuitive sense that a crushing injury, so let's say a finger or a hand has been crushed by some type of mechanism. A crush injury allows a more full recovery to an axon than a complete cut, so then a complete sever to a nerve, which sort of makes sense. The axon's still intact and a major crush injury, so sort of greater hope of regeneration than if an axon gets completely whacked. I will tell you though, this process, even if it will sort of regrow, is extremely slow. So I don't know if you guys remember at the beginning of last semester, I shared with you the story about my husband putting his hand accidentally through the pane of glass in the window. And I was showing you as an example of a really nice first intention, sort of primary intention healing of that scar in his hand. I think I also mentioned to you too, during that injury though, he did cut his ulnar nerve. Did I share that with you as well? So he had to go have surgery. There was actually still a little tiny piece of glass from the window in his hand still that had sort of gone down into his hand and sort of cut his ulnar nerve kind of right here at the sort of hand-wrist junction. And the hand surgeon, the neurologist said, there's a pretty good chance that you're going to have some sort of feeling, because his ring finger and his pinky finger were numb as soon as it happened, so we knew something happened with the nerve. He said it's a pretty good chance that you're going to get some sort of function back, but it's going to be super slow. We won't know for like 6 months to 2 years how much sensory you get back to those two fingers. And he shared with us it's really like 1 millimeter a month. So it can be really slow, and especially the injuries down here. So we're talking 6 or 7 months, 6 or 7 millimeters, so to speak, before we know how much sensation he gets back in these two fingers. So again, though, it all the whole, and it is maybe sort of coming back. He's not sure. Nerve injuries are really weird in that you don't know like how you're feeling. One day it's burning, the next day it's throbbing, the next day it's super sensitive to cold. So when I ask him, so how are your fingers doing lately? He's like, I'm not really sure. They feel funny, which I think is a good sign, that they feel something. But anyway, it's this perfect example of Wallerian degeneration and what happens to that axon when it gets cut, and then what happens when you try to realign and actually have it kind of start to regrow. Yeah? Say that again. Sort of, yes, sort of. Kind of a phantom limb sort of pain in that, now that it's starting to grow back, hopefully, that it's painful. There's a lot of sort of burning sensation, a lot of pain in that area. And again, I think it's a good sign that he's having some discomfort, because at least that means he's getting some sort of innervation down that axon root. Good. And here's a perfect example of that. So let's say this is his example of his ulnar nerve, right? So it gets cut, and as it starts to try to regrow, you can see here, when cut, a damaged motor axon can regrow to its distal connection only if the Schwann cells sort of remain. So here's the Schwann cells, these yellow, part of the myelin sheath along this axon. They start to regrow, but see how funky they start to regrow? They're totally unorganized, which is, I think, one of the reasons why he has such a strange sensation going on right now, because it's there, it's just wackadoo, right? It's not making any sense right now. And then as it starts to sort of realign and get more mature, it does kind of start to realign and go through that column that those remaining myelin sheaths have formed and innervate that effector muscle, or in his case, the sort of function of his ring finger and pinky finger. Yeah. 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 That's where the cut was. Yeah. So from here, so here's the cut. So from here down would be where the Wallerian degeneration would be occurring. So distal to the injury, distal to the cut. Right. So here's the degeneration occurring, and you can see how the muscle here is actually starting to atrophy because it's not getting sensory innervation, or innervation period, I should say. And then as it starts to sort of regrow, it regrows funky, and then hopefully becomes a little bit more organized, a little bit more mature, and actually does provide a guiding tunnel in which that axon to sort of regrow back to its effector organ. Does that make sense? Okay. So any questions about structure before we move on to sort of the actual sort of messaging, synapsy kind of conversation regarding nerves? Okay. So how does it work? What conversations occur on these nerves? We talked about the structure. We've talked about sort of the function, so to speak. But what conversations, what communication actually occurs along these lines, along these phone lines, so to speak, that run throughout our entire body? So we know, right, that an unexcited neuron maintains a resting membrane potential. We talked about that ad nauseum last semester. When a membrane potential is sufficiently raised, and by the way, how is a membrane potential sufficiently raised? Sodium arrested into those cells. Beautiful, good. You didn't forget everything over break, right? Good. So sodium coming into those cells, and action potential then is generated. So yay, good. We want that to happen on nerve cells. And the nerve impulse then flows down that axon to the next neuron, to all the parts of that next neuron across that synaptic gap. So very good. So as you know then, neurons are not physically continuous with the next neuron, right? So what is this communicating factor between neuron 1 and neuron 2? Synapses, neurotransmitters, right, exactly. So we know that synapses are that region between adjacent neurons. Impulses are transmitted across that synapse by two different types of conduction. So by chemical and by electrical conduction. So on a presynaptic neuron, so here, here is my little, okay, this is the picture we're looking at now. I know I've got tons of different pictures going on, so let me just kind of highlight it here for you. Here's the picture we're talking about now inside this box. So here's my synapse. Right here, right? Here's one neuron, axon, synapse, second neuron, so dendrite essentially over here. So we've got our presynaptic neuron here, and we've got our postsynaptic neuron here. So conduction between this neuron and this neuron happens in two ways. There's a chemical or electrical conduction down the axon, so we know this is the electrical sort of action potential that we talked about. And once it gets here to the very end of the axon, when it reaches the synapse between neuron 1 and neuron 2, we now have a what type of communication across that synapse? Electrical or chemical? Chemical, because of the neurotransmitters. Good. So the presynaptic neurons are the ones proximal to the synapse. The postsynaptic neurons are the neurons that are distal to that synapse in terms of that relationship. And it's the presynaptic neurons that we're talking about, the electrical component of the relationship. And then it's here in this synapse that we're talking about the chemical relationship, part of the relationship. Does that make sense? Okay. What are the chemicals? Yeah. Mm-hmm. Yes. Yes. So it's basically the it's basically the electrical sort of action potential that moves down the axon of that presynaptic neuron that actually that sort of activates and excites those little synaptic boutons that hold the neurotransmitters at the end, and excites them so much that they release their chemicals, they release their neurotransmitters into that synapse and then get attached to the dendrite. Yes. Well, so good question. So once it gets here to the dendrite, right, to the dendritic receiving portion of the next neuron, that basically stays chemical until it gets, so it moves from the dendrite into, so through to the body, and then what happens at the body is that it creates that sort of action potential because it changes, exactly, so it changes that action potential in the cell body, in the soma, that activates and sort of excites that axonal hillock where the action potential begins and starts it all over again there as electrical. Does that make sense? Okay, was that your question too, sort of? No. Capable of both? Yes, yes. Because basically from the dendrite to the cell body, it's chemical because it's received the neurotransmitter, and then the neurotransmitter then excites and changes the action potential, allows the sodium to rush into those cells, which changes that membrane potential and makes it electrical again, and then that electrical impulse shoots down the axon. So yeah, basically if you want to think of the dendrite and the cell body as basically being sort of the chemical aspect and the synapse, right? The synapse, the dendrite, the cell body, and then it changes into electrical once it changes the action potential of that axonal hillock, and then it becomes electrical again until it hits that synapse. Yes? Makes sense? Okay. So we know that the chemical aspect of this relationship comes from neurotransmitters. There's more than 30, and we know that they can either be excitatory or they can be inhibitory. Anyone want to sort of take a stab at remembering what sort of an inhibitory neurotransmitter is? What's the name of an inhibitory neurotransmitter? Do I remember? Serotonin. Serotonin's a big inhibitory neurotransmitter. So whatever it's sort of being sent to would actually inhibit or sort of slow down whatever it's being sent to. An excitatory example of neurotransmitters would be like norepinephrine, adrenaline, sort of epi, norepi sort of things, to sort of excite and sort of stimulate whatever it's being shot to, so to speak. And then I mentioned these synaptic boutons where neurotransmitters are stored within a presynaptic neuron. So they're sort of stored right here at the end of this axon. So let's say we're talking about serotonin. The neurotransmitter serotonin is stored here in these little pouches. And then when it gets electrically stimulated, it releases it out into the synapse to be picked up and received by this dendrite. And actually, here's a great picture of that. I'm going to move forward here just for a second. So right here, don't get too hung up about all of this stuff that you see in this illustration. What I want you to really focus on is this synaptic relationship down here. So here are our little synaptic boutons that are sort of housed right here at the end of this axon. It gets electrically excited down the shaft of this axon, releases these synaptic boutons to release, or excites the synaptic boutons to release the neurotransmitters, and then it jumps into this synapse and goes over to these receptors here on this next axon, or on this next neuron on the dendrite. So interestingly though, and I hope it sort of makes sense after what we just talked about, chemical synapses operate in one direction, right? Because the neurotransmitter is normally stored on one side, on the presynaptic axon, and then released to this postsynaptic neuron, or this postsynaptic synaptic dendrite. So it's a one-sided relationship, right? And it can happen by sort of a term called summation. And it can either be either temporal or spatial. So temporal summation, well, actually, let me take a step back and sort of describe something else for you before I go into this. So usually, a single excitatory postsynaptic potentials cannot induce a neuron's action potential and sort of the propagation of that nerve impulse from the dendrite down into the cell body through the axon like we talked about. Whether this occurs, whether that action potential and that whole sort of excitatory aspect of a relationship in that neuron occurs is due to the number of frequency, or I should say the frequency of impulses it gets from that presynaptic axon. Does that make sense? So it can either be temporal, so the effects of successive rapid impulses received from a single neuron at the same synapse. So it just keeps, one neuron just keeps exciting that neuron number 2 until it finally reaches an action potential and allows for that impulse to be propagated down the cell. So it's a time relationship, and by time I mean the number of times a neuron gets excited from the presynaptic neuron. Or spatial. So spacing effect would be the combined effects of impulses from a number of neurons onto a single neuron at the same time. And actually, spatial summation is the perfect example, or basically the definition of convergence. So the only reason this neuron is getting excited and propagating a nerve impulse is because it's being activated by several different neurons, and it finally reaches that sort of action potential, what's the word I want to say, sort of level in which it has to get to actually get excited. It takes that many neurons to get it excited. So if only one of these neurons was trying to excite it, it wouldn't work. But because it's being fed and activated by 3, it does propagate a nerve impulse. Does that make sense? Maybe, sort of. Yes. So in the example of temporal summation, it's just how many times one neuron is firing at another neuron, and once it reaches a certain number, it creates an action potential in that first neuron, or in the second neuron. In spatial summation, it's not how many times it's being activated, it's how many neurons are activating it. Does that make sense? Okay. All right. Whew. Any questions about that? We're going to get to a little bit more straightforward conversation here when we start talking about actual structures and functions of the brain. So any questions about sort of the messaging chemical aspect, synaptic aspect of the neuron? Okay, good. Good. Now we're going to get to the more sort of easy to visualize fun stuff. Okay, so whoopsie. So the central nervous system, we're totally switching gears here now, right? We're getting away from sort of that functional stuff, and we're talking more now about structure of the central nervous system. So central nervous system is made up of, obviously, the brain. Hello. And it contains the forebrain, the midbrain, and the hindbrain. So forebrain is formed by the 2 cerebral hemispheres, right? And we know that the cerebral hemispheres are made up of 2 different types of matter, gray matter and white matter. We know that the brain is approximately, the brain itself, so I'm kind of taking a step back here away from the forebrain and talking about the brain itself. The brain itself is approximately 3 pounds. It receives, now listen to this, 15 to 20% of the total cardiac output. So the amount of blood that our heart pumps out to reach all the different organs and all the different areas of our body, the brain takes 15 to 20% of that. That's a lot. So that tells you how dependent on blood supply the brain is. It takes basically almost a quarter of our cardiac output for itself. That's huge. And we'll talk more about that as we get farther into the neurologic conversation. But it takes a lot of our cardiac output. Okay, now we'll go back to the forebrain. So formed by the 2 cerebral hemispheres made up of gray matter and white matter. The cerebral cortex contains the cell bodies of the neurons. So cerebral cortex as part of those cerebral hemispheres. What makes gray matter gray and white matter white? Anyone know? I think I heard it. The myelin sheaths, right. So if the sort of myelinated part of a neuron, we know, is the axon. So that part, the white part, is the myelinated part. The gray part is the unmyelinated part. So now we're talking about the cell body, basically, is the unmyelinated part that makes up the gray matter. So basically what I want you to take away from this is that the forebrain, the two cerebral hemispheres, contains both gray matter, so the cell bodies, and also white matter. So it also contains axons. So it contains the cell bodies and axons in its components in the cerebral hemispheres. So the cerebral cortex contains the cell bodies, so the neurons, of the neurons, the gray matter, and the white matter lies beneath sort of the cerebral cortex. So it sort of lies underneath that. So if you want to think of it simplistically, the white matter is sort of more internal, and the gray matter is sort of more on the outside, sort of outside layer of that forebrain. Mm-hmm. Yes. And it should make sense to you because the white matter, what's white is the myelin. And so then obviously we know the myelin is on the axons, right? Okay. Okay, so then we also have the midbrain. And then we have a portion of the brain called the hindbrain, which contains the cerebellum, the pons, and the medulla. Interestingly, you see here, this is at the bottom of the screen, the midbrain, the medulla, and the pons make up something called the brain stem. Now, why is the brain stem so important? Why are we so interested and concerned about the brain stem? Okay, so first of all, the brain stem connects all of the other parts, all of the other structures of the brain. So it connects the brain, the cerebellum, and I should say the brain, the cerebrum, the cerebellum, and the spinal cord. So it connects everything together. So that's one reason that the brain stem is super important. What other things occur? What other sort of functions occur in the brain stem? Okay, let me ask you specifically in the medulla. So the medulla is part of the brain stem. In the medulla, what occurs in the medulla? Anyone remember? It's where our motor pathways cross. So that's a big deal. Our motor pathways cross in the medulla, which is included in the brain stem. And what functions does the brain stem have? What are we so concerned about in terms of the functions that the brain stem sort of controls? Yeah, so the brain stem affects those very basic life-supporting functions. So heart rate, breathing, sleep-wake cycle, so ability to sort of be awake and be conscious, eating drives, things like that. So that is why we are so concerned with the brain stem. If we have a patient with a major sort of traumatic brain injury to the brain stem, or let's say they have a stroke, they have a cerebral vascular accident in the brain stem area, we're very concerned that significant damage to the brain stem is not compatible with life because of the issues with vital signs, heart rate, breathing, the ability to sort of be awake and stay awake. So we're talking about patients in vegetative states where they're in this sort of unconscious state. Again, probably some type of injury or infarct to the brainstem if they're not able to sort of maintain normal wakefulness. That's why we're so concerned about injuries to the brainstem. Also, interestingly, 10 out of the 12 cranial nerves have nuclei embedded in the brainstem. So also very important in terms of sort of bodily functions of the rest of our body as well, because a lot of those cranial nerves are housed in the brainstem. So hopefully that should get across to you the importance of how important the brainstem is. Here's just a picture of the brainstem sort of after autopsy. So brainstem, pons, medulla, and then you can sort of see a little bit of the midbrain here. The reticular formation is also another really important part of the central nervous system. It's the network of connected nuclei that regulate also vital reflexes, such as cardiovascular and respiratory functions, so heart rate and respiratory rate. Together with the cerebral cortex, it's referred to as the reticular activating system. And again, what I really want you to remember about the reticular activating system is that it's essential for maintaining wakefulness. So the reticular activating formation is essential for maintaining wakefulness. So again, injury to the reticular formation or the reticular activating system causes basically an unconscious patient, a vegetative patient that cannot maintain and sort of achieve wakefulness. So again, these are not just structures that you memorize and sort of think about in terms of like anatomy and physiology, but I'm trying to bring them back to the real world for you and let you know why we're concerned about them, why we want you to know about them. Here's just an illustration of sort of the things that we've talked about and sort of how they all connect. Reticular formation, thalamus, those types of things. Okay. All right, the forebrain, quickly, contains something called the telencephalon, which contains the cerebrum and the lobes of the brain. So the cerebrum, we know, contains the gyri, which are sort of the convolutions, those, you know, the parts that makes the brain look sort of wrinkly. The purpose of the convolutions of these gyri are to increase the surface area and to increase the number of neurons that can be housed in that area. The sulci are the grooves, sort of the valleys of those folds. The fissures are the deeper grooves, basically, is the way you can think of it. And then we have the lobes, actually. So we have the frontal, the parietal, the occipital, and the temporal lobes. Tell me about what you know about the functions that sort of are housed in each one of these lobes. So what is the frontal lobe sort of known for? So personality, good. What else? Learning, sort of cognitive abilities, good. In terms of cognitive, more along sort of executive functioning, so ability to sort of reason, do math, sort of in your nursing realm, sort of like your ability to sort of critical thinking, you know, those sorts of things. Also inhibition, and this is really interesting. When I think of the frontal lobe, I always think of inhibition, or the lack of, you know, therefore the lack of inhibition, because when I used to work with neurology patients at Penn, there's a specific type of dementia called frontal temporal degeneration or frontal temporal dementia that is just basically degeneration of the frontal and the temporal lobes, thus the name. But what's really interesting is these people they look totally normal, they can hold jobs, they have families, they have houses, they pay their mortgage, they can be almost basically perfectly functioning members of society, but they lack inhibition like no other. So these are men who, you know, are patients. I'm thinking of one particular guy who is a totally normal functioning member of society, but like goes out and does these crazy weekends of gambling and extramarital affairs and speeding and DUIs and just these crazy like lack of inhibition sort of behaviors. And it actually turned out it wasn't just his sort of behavior. He actually had frontal temporal degeneration. So when I think of inhibition, I think of frontal lobe. Really interesting. Parietal lobe, what's sort of housed in the parietal lobe? Anyone know? Sort of like somatic sensory is the big function of the parietal lobe. Occipital, primary vision, visual, good. And then temporal. So as I mentioned, in frontal temporal degeneration, primary auditory, language, and then also an area of the brain called Wernicke's area is in that area as well. Anyone know what Wernicke's area is sort of responsible for? Interpreting speech. Very good. Very good. Interpreting speech and understanding speech in your environment. Being able to sort of comprehend what's being said around you. Perfect. So obviously dysfunction in that area may result in a specific type of aphasia called receptive aphasia, and that aphasia basically means difficulty speaking, and so obviously if you can't understand what's being said around you, you're going to have trouble talking, right, because you have no idea how to comprehend what's being said to you. And so that would be something, if there's damage in that Wernicke's area within that temporal lobe, would be a perfect example of that. Good, very good. So actually that brings us to my next slide, which is we're going to talk more about Wernicke's area dysfunction. So the forebrain continued. So the forebrain not only contains an area called Wernicke's area, it also contains an area called Broca's area. And they're very different areas, but they're both housed in the forebrain. So Broca's area is on the left hemisphere, the left side of the brain. It's responsible for motor aspects of speech. So that's to say that hopefully my Broca's area is working very well, because hopefully the words that are coming out of my mouth right now, you're hopefully halfway understanding, halfway making sense, that I am able to sort of know what I want to say and actually say it, actually spit it out, and make sense of it so that other people can understand. It's also sort of the motor aspects of my speech in that I can talk in sentences. I'm not just standing up here trying to get my point across in one word sort of phrases. That's what Broca's area is responsible for. Responsible for the motor aspect of the way I talk, the motor aspect of my speech. As opposed to Wernicke's area, so if I had let me put it this way, if I had an issue with Broca's area, I'd be standing up here saying, brain, good, works good, right? Literally, I know what I want to say, but I can't get it to spit out. It's very choppy. The motor aspect of my speech is not working. I can't get my point across to you because I just physically can't make the words with my mouth. As opposed to Wernicke's area, it's a sensory speech area, as was mentioned. So what that means is that this area is responsible for sort of reception and interpretation of speech. So you could be talking to me about your winter break, and I could be sitting here nodding my head like, yeah, totally, sure, sounds fun, but really I have absolutely no idea what you're saying. So then when it's time for me to talk and me to sort of reciprocate in the conversation about your winter break, what comes back out to you is complete gibberish. Because how can I make a meaningful statement to you about your winter break when I don't understand a word you said? So what's really interesting about dysfunction in Wernicke's area is that I could be talking up here, and if you put me on mute, it could look like I was perfectly normal. I could be like, I could be talking, and if you put me on mute, you're like, oh, she knows what she's saying. But if you actually turned up the volume on what I was saying, it would be complete gibberish. I mean, just nonsensical words and gibberish. Because how can I talk if I don't understand what was just being said to me? Okay? I could stand up here and talk until I'm blue in the face about these two differences, but I think it's going to make more sense to sort of visualize it, and I've got some YouTube videos here for you. So the first example that I'm going to give you of a dysfunction in Broca's area. So what you see here on the screen is what should happen under normal circumstances in Broca's area. But what you're going to see here in the video is a dysfunction, is some sort of infarct or injury to Broca's area and what that does to your speech. Let's hope the volume is good enough. That's what I was checking at the beginning of class.
Arizona Congresswoman Gabrielle Giffords has stepped back into the spotlight, given her first interview since she was shot in the head in January. 2020 gave us a first look at how far Giffords has come. For the first time, we learned that her husband, Mark Kelly, had been videotaping his wife's progress all these months. The images of Giffords early on in her recovery are certainly sobering. What a contrast they made to this.
How do you feel?
Pretty good.
I can see that your arm, your right hand, you move a lot more. Now, is it painful? Is it hard? No, it's
difficult.
Just difficult.
Difficult.
Yeah.
Difficult. Strong.
Okay.
Knows what she wants to say.
That's the spirit,
She's got
isn't
very
it?
good posture,
She
much better than me.
get it out.
You do a lot of therapy every day.
Yeah. How many hours?
2 hours of therapy.
Here at the house.
Yes.
Giffords has clearly come a long way. She looks amazing. Her spirit, energy, intelligence, all intact. But it's also obvious she has so many thoughts she's unable to express. She speaks mostly in single words, not in sentences. Let's talk about that with Chief Medical Correspondent Dr. Sanjay Gupta. He's a practicing neurosurgeon as well and sees a lot of patients like Gabby Giffords.
Okay, so that, like I said, I just wanted to show you an example. She is, in terms of her injury, she is totally aware of what's going on around her. Her intelligence has not been impacted. It's literally that she knows what she wants to say and she can't say it. Now, can you imagine how incredibly frustrating that would be? Yeah. Yes. So it's all speech. So she could write. She could write, she could listen, she understands what's going on around her. It's the actual motor aspect of her speech. And if you've heard her talk, I don't know if any of you have sort of heard her or seen other interviews, She just really perseverates. Like you can tell, you know what she wants to say, she just can't get it out. And so, you know, like when she said, "How are you doing?" I'm sure she could tell you a book of how she's doing, but all she can spit out is good, strong, strong, you know, getting better sort of thing. That is the perfect example of Broca's speech. It's an expressive aphasia. She can't express herself. That's the perfect example of expressive aphasia. Now, here's another example of a disorder with Wernicke's area. This is a video, I don't know if some of you might have seen this several years ago again, of a local reporter in LA who was covering the Grammys, like live at the Grammys, and had some sort, they actually now blamed it on a sort of strange effect of a migraine. So it isn't necessarily a true infarct in Wernicke's area, but it affected, for some reason, the Wernicke's area and affected her speech on live TV. So let me show you this picture. And again, show you kind of the example of, looks fluent, but complete gibberish.
Highlights and backstage coverage we're seeing for the very first time, Sari.
Well, a very heavy rotation tonight. We had a very Darrison. Let's go ahead, Terrace Jason. Let's put it at the.
So again, not necessarily a true infarct or a true stroke to Wernicke's area, but something happened with this migraine to impact that receptive interpretation of her speech. And so again, if you would have muted her, right? Hold on, guys. If you would have muted her, she would have looked totally fluent, right? Standing up there. But what was coming out of her mouth was complete gibberish. That is an example of receptive speech, or I should say a trouble with receptive speech. So I just want to kind of give you an example there of the two differences and how they look. Good. So that's just kind of what I wanted to end there. I'm going to keep going here just to kind of get a little bit more in before we have to stop. I'm more than happy to answer some more questions after class at the break. So again, here's just kind of a visualization of a visualization of the forebrain and the different lobes. This slide, in terms of talking about the next sort of area of the brain, diencephalon, I'm not going to spend a lot of time on this. What I do want you to focus on are two things, the thalamus and the hypothalamus. The thalamus, which we're going to talk about later, and we're not going to get to that diagram over there on the board, which is fine, because we're going to have time to do it on Monday. But the thalamus is the major relay station of sensory information. So any information that we receive from our environment comes up through our sensory system and gets housed in our thalamus, and then the thalamus is sort of the relay station to move it on up into the sensory cortex of our brain. So when you think of thalamus, think relay station. And hypothalamus, which again, we're not going to talk a ton about today, but it is responsible for a couple of things. Maintaining sort of internal environment. So next week when we talk about sort of the brain's control of our bodily temperature and like fever and things like that, the hypothalamus is the big thermostat of our body. And that's what we'll talk about next week. And then also the hypothalamus also plays a big part in endocrine function, and we'll talk about that when we talk about the endocrine system and endocrine system dysfunction later on in the semester. But those are the two things I really wanted to sort of focus on that slide. Okay, the hindbrain, we know that the cerebellum is what? What's the cerebellum responsible for? Movement, actually more coordinated movement. The ability to sort of hold posture, be coordinated in fine little motor movements, things like that. Balance and posture. The pons aspect of the hindbrain, when you hear the word pons, I want you to think bridge. Bridge. It transmits information from the cerebellum to kind of all the other areas of the brain. So to the brainstem and to those two cerebral hemispheres. Just any of the information. Transmits. It transmits information from the cerebellum to the brainstem and to the cerebral cortex, or to the cerebral hemispheres. So think bridge. All right. And then the big thing that I want you to focus on in terms of the myelencephalon is the medulla. This is sort of the lowest portion of the brainstem, but again, it is part of the brainstem. So you really, when you think of the medulla, think of all those functions of the brainstem that we've already talked about in terms of heart rate, respiratory rate, blood pressure, even things like coughing and sneezing, all those sort of like reflexive things that we do that we don't really have to think about and sort of try to control ourselves occurs in the medulla. And what other really important function or aspect of our neurologic system occurs here? Anyone know? The corticospinal tracts cross here. So that's why the left hemisphere controls the right side of the body and the right hemisphere controls the left side of the body, because they cross here at the medulla. Yeah, the corticospinal tract conducts impulses from the brain to the spinal cord, and it actually crosses, as it's moving down from the brain to the spinal cord, it actually crosses in the medulla. So that's why the left side of, or the right side of our brain controls the left side of our body, and the left side of our brain actually controls the right side of our body, because things cross there in the medulla as they come down. Does that make sense? Okay. All right. So now we're going to move from the brain and move down a little bit further and talk now about the next part in the chain of command, which is the spinal cord. The spinal cord, you know where it is. You know that it connects the brain to the body, so to speak. It conducts both somatic and autonomic reflexes. And then you see a couple of the other sort of functions there on the screen. But again, you know that, basically. Again, the other thing that's really important to understand about the spinal cord is that it also, just like the brain, contains both gray matter and white matter. So that tells you that it contains both the cell bodies of a neuron, but also contains some of the axons as well, because it contains both the unmyelinated, which is the gray, and the myelinated, which is the white matter. The gray matter, and this is your question that you had earlier, Kathy, the gray matter within the spinal cord is in a butterfly-shaped inner core inside the spinal cord. So in here is the gray matter inside the spinal cord. And the white matter sort of surrounds the gray matter all the way around the rest of the spinal cord. So that's where you see those two different types of matter being housed in the spinal cord. Within the white matter, so on the outside, which surrounds the gray matter, within the white matter is where you see both those ascending and descending pathways that we talked about at the beginning of class today. That's where you see them housed. And we call them spinal tracts, so they're housed here on the outside. We know that the dorsal area controls the sensory, and actually, to kind of make it very simple, the ventral area, or the front area of the spinal cord, right here, is what controls more of the motor, sort of voluntary aspects. So that being said, you have the spinal thalamic tract and the corticospinal tract. Now, to make it easier for you, the way that these tracts are named denotes where they begin and where they end. So that being said, if you hear the term spinal thalamic tract, is that an ascending or a descending pathway? Ascending. It's carrying information from the spinal cord, so spino, up to the brain, up to that relay station, the thalamus. So spinal thalamic tract is ascending and carries sensory information, as opposed to corticospinal tract carries information from the cortex, the brain, down to the spine. So that is a descending tract and carries what type of information? The motor, and would be found where? The ventral side of the spinal cord. Yes? Does that make sense? Okay. All right. I think we're actually going to stop there today. And we will finish up on Monday talking about the rest of this information, and begin talking on Monday again about other sort of aspects of neurologic function in terms of body controls, like thermoregulatory sort of temperature control and things like that. Okay? No, I'm not going to do a voiceover because we're going to have time to do it on Monday.